Okay, so hello all. Welcome to the first online session of developmental biology. Just a minute. I think students are joining. I think up till 10 to 15 minutes they'll keep on joining. Okay. So I am Apurva Soni. I am a PhD scholar in University of Hyderabad and I will be your teaching assistant for the rest of the session. And so basically the major objective of this online session would be to solve some sample questions which will help you to understand your assignment questions and which will help you to solve your assignment questions. So we'll be solving some 10 to 12 questions in this 2 hours, and as well as we'll try to just briefly quickly summarize or just recapitulate some of the points which are important uh, of that week. Okay, so I think your week 1 and week 2 both online lectures are now available. So today we'll be starting with week 1 online lectures. So we'll be solving questions related to week 1. So I just wanted to know that have you gone through the lectures of week 1? Have you gone through the recordings?
Yes, ma'am.
Uh Nabandita, I couldn't hear your
Yes,
voice.
ma'am. Yes, ma'am. I have gone through the lectures.
Can you just type? I don't know whether there's a problem at my end or um. So can anybody else hear her voice? I just wanted to know whether there is a problem at my end or okay, fine. That's really good that you have gone through the lectures. So my suggestion and my request would be that okay, yeah, fine. That's fine. It's okay. I think there is some network problem. That's fine. So my suggestion would be that before coming to the class, if you can just go through the lectures, okay, so it would be beneficial for you only because then you would be able to understand the concepts or whatever questions that we'll be solving a lot more easier. So, as I was telling that my name is Apurva and I am a PhD scholar at University of Hyderabad. And because I have a fellowship which is PMRF, so for that fellowship we have to do a teaching assistantship. So that's why I'll be teaching you for the next whole 3 to 4 months. So just quickly, can you tell me that you guys are like bachelor students or master student at what level? So that I'll try to make you understand the concepts in that. Yes, Shweta. Yes, you wanted to say something. You can say.
I'm sorry, ma'am. I clicked it by mistake.
Okay, I think there is a problem at my end only because I am unable to hear anybody's voice. Just a minute. Okay. So Shweta, can you say it again? I just wanted to check whether there is a problem at my end. Is it resolved? Or then otherwise you can
Can you hear me, ma'am?
ha now I
Hello?
can hear you. Yes, I can hear you now. Yes.
Yes, ma'am. I clicked the button by mistake, ma'am. Really sorry.
It's fine. Hmm. So you are bachelor student or a master student? Shweta, you are a bachelor student or a master? Okay, master student. Fine.
Ma'am, I'm a bachelor student. I'm doing a BSc Biochemistry, ma'am.
Okay, okay. BSc Biochemistry. Good. So Soumya has replied she's a master student. Devnath has replied that master student. Okay, fine. So I think now we'll start the lecture. So for some minutes, I won't get go to the slideshow because some students might join the class. That's why I have to open this one only. Ananya is replied bachelor student. Okay, fine. So before starting the questions, just having a basic concept of what developmental biology is. So, as we all know, that biology is a very vast area, a very vast topic, and it encompasses a lot many fields. Okay, so when we talk about biology, it's about learning the molecular mechanisms that keep our body alive. It's about learning about the cellular physiology. It's about learning of the plant system. It's about learning of the animal system. And along with all these questions, one major question or one major area, which is indeed one of the most important area of biology, is that how an organism evolves, or how an organism grows from an embryo to an adult. It's, I think it's one of the most phenomenal uh, process that occurs in biology, that how a single small egg, when it gets fertilized with a sperm, how that egg develops into a whole complete organism, and how the process is so very well maintained that even a small division mistake, or even a small replication mistake, or even a small mutation can lead to such a substantial change in the formation of the body. Okay, so for example, some people have six fingers, or not just five fingers, six fingers, or some little bit abnormalities. So this is because of some of the errors that has occurred during the development from the embryo up to the adult. And if you have gone through the lectures, one, so sir has explained things, sir has explained it very beautifully, that any machine, if there is any machine, it has to work. So by the time that machine gets completely made, it is functional, it cannot work. But when we talk about living organisms, so the basic idea of the life cycle of a living organism, or a basic idea that what is the uh, threshold point when we can say that a particular organism or a living being is functional, is when that organism or a being is reproductively active. So when I talk about the life cycle of an organism, always remember there is a difference between life cycle and generation time. Okay, there are two terminologies. One is life cycle, and the other one is generation time. Fine. So generation time is the time from birth till death. That is your generation time. But when we talk about life cycle is the time span from birth up till the organism is reproductively active. So that is your life cycle. So when we study about life cycle, we are studying the whole processes when an organism gets reproductively active and it can reproduce. But for example, for example, humans. So by the time the human gets puberty and it gets reproductively active, even before that the human being is functional. All the organs are working. He does work. He does go to the school and everything. So this is a beauty of our living system, that even before we achieve the life cycle complete maturation stage, the organism is functional, right? So if we define what is developmental biology, so what is developmental biology? It is the science that investigates the process of organism's physiological and functional maturation over its life cycle. That means how from a single celled egg, how an organism develops into a fully functional organism that is reproductively active. So the study of this transition is done in developmental biology. Okay, and it explains the variety of interacting processes that generate an organism's heterogeneous shape, size, and structural features that arise on the trajectory from embryo to adult, or more generally through a life cycle. So when we say adult, adult means when the stage when an organism is reproductively active, that is known as adult. So this is a basic definition of what is reproductive biology, or sorry, what is developmental biology. So now I think I'll start with the questions. So with the questions, we'll try to cover the topics that are being asked in the questions. Fine. I think Okay, so Ajay has asked a question. Conception to birth is part of life cycle, and okay, I think there are many people who wants to join the call. Just a minute. Okay, two people. Fine. Yeah. Okay, Ajay. So the basic difference between generation time and life cycle, as I already said, that an organism's birth till death, that span of time is known as generation time. Okay. So, for example, up till 62, if an organism, if a human is living up till 62 or 70, so up till 70, that time is this time is generation time. But up till 70, is that organism or is that human sexually active or reproductively active? No, it will be active up till a particular time. So the time span that a human takes or an organism takes to get reproductively active, that time span is known as its life cycle. Okay, because there are many organisms uh, which we'll see eventually that they spend most of the part of their life being sexually inactive, and as soon as they reproduce they die. So their sexually active portion of their life is very less. Most of the time of their life they are sexually inactive, and when they become sexually active they reproduce and then they die. So that's why, Ajay, can I use mic? Yes, you can. If you have anything, if you have any question, you can. And when I'm explaining, try to keep yourself mute so that none of the students gets
Madam,
affected.
madam,
Yes.
you said कि life cycle is from birth to reproductive life,
Hmm.
and generation time is from birth to again
Death.
is able to generate another generation.
No.
Generation.
Generation time is from birth till death. But life cycle is not from birth till death. Life cycle is from birth up till the age when an organism becomes sexually active, reproductively active. That is your life
Then,
cycle.
but in the life, from conception to birth, that is also part. So that part is in the life cycle or in generation time. That's what.
Can you repeat? Your voice is not very clear actually.
From conception to birth, it is part
Hmm.
of life cycle or it is a part of generation time.
No, it is a part of life. It's a part. See, uh,
You have
generation
excluded
time
this.
is a broader perspective. It's
Well,
a
agree
broader
with
area.
generation.
Generation time will include your life cycle time and plus the additional time which you live after you complete your reproductively active age also. So that will be your generation time. So you try to understand it in such a way that life cycle is a small portion within the generation time. Could you understand this?
I am able to understand what you are saying, but what my query in my mind is generation is something a process, means active process. So generation life cycle is from birth to death. Generation time is means from birth to maturity.
No.
That's what my
Okay,
understanding was.
first you need to
If
clear. I think you're
you
getting
can confirm.
confused, Ajay. Let me speak, please. You said completely the opposite of what I just said. Okay, you are saying that life cycle is from birth to death, but I'm not saying that. Life cycle is up till the time span from conception. Like for example, an embryo. As soon as the embryo gets fertilized, okay, from that time up till the time that particular organism becomes an adult, that is, it becomes sexually mature.
Why
That
embryo should get fertilized? Ovum should get fertilized.
yes, ovum should get fertilized. Otherwise, it's just an egg.
Embryo is the product of conception we called embryo. No?
Yeah, so yeah,
Before
that
that,
is
it
what
was
I'm
a
saying.
product of conception.
Can you just type it because I think your voice is not very clear. Okay, so I think Ajay is typing. Okay, fine. So I think we move. Otherwise, we have to finish the questions also. I think Ajay is typing. Fine. Whenever he'll type, will try to. Okay. Just a minute. So the first question is, KIT gene is expressed in which of the following cells? Presumptive pigment forming cells, precursor of blood cells, germ cells, and cardiac muscles. I'll give you 2 minutes time. Then I'll switch to then I'll switch the screen.
Mm,
I think if you have the answers, you can wait for 2 minutes. Let other students also think and answer. And after 2 minutes, you can type in the message box. After 2 minutes, I'll switch the screen and then I'll see the message box. Okay, so that everybody, we have to take everybody with the same pace. So if you know the answer, well and good, you can type it after 2 minutes. And if you do not know, you can think. Okay. I'll switch the screen and I'll see what you have answered. Okay, let me see. A and B. A and B, A and C. All, if allowed, germ cells. Okay, the question is, KIT gene is expressed in which of the following? So, many students have answered A and C, A and B. Okay. So the correct answers, so if you have answered either of them, it is correct. Presumptive pigment forming cells, precursor of blood cells, and germ cells. Okay. So what we'll do is, I think it was in lecture 2, I guess, about KIT genes. Lecture 2 or lecture 3. Mm. So we'll try, like whatever questions we are dealing with, we'll try to see a little bit about the questions. Yes, here it is. So I think many students have answered the question correctly. Germ cell is also correct. A and C. What is A and C? Okay. Yes. So the only incorrect statement in this question is I think cardiac muscle. A and C, A and B. I think most of you have answered it correctly only. Good. That means you have read it. So you can see that KIT gene is expressed in neural crust cells, it is expressed in precursor of blood cells, and it is also expressed in germ cells. So basically, what does this KIT gene tells us? So there are certain kinds of birth defects that happens during the human development, and this defect is because this birth defect, you can see here, you can see here is a patch. You can see this kind of patch occurs, and this syndrome is due to the mutation in the gene which is known as KIT gene. So such children who have this particular disease which is known as pilbardism, so such children do not survive for a very long time. Okay, so if we have to study about this gene, obviously we cannot use a human model because they do not survive for a very long time. So this particular syndrome is also observed in your mouse. So mouse can be used as a model organism for the study of this gene. Okay, so basically when we are dealing with the study of any kind of genes, or we are dealing with the study of any kind of syndromes which are happening because of mutation in certain genes. If possible, we try to take a animal model. First of all, our first approach is always to do in vitro. So can anybody tell me the difference between in vitro and in vivo? What is the difference between in vitro and in vivo? Anybody? Anybody who knows the difference between in vitro and in vivo? In
In
body.
vitro is something that takes place outside the body.
Yes, that is known as
In vitro,
in vitro. Yes. And in vivo is that occurs inside the body.
yes,
Yes,
ma'am.
correct. So whenever
Yes,
we are
ma'am.
trying to understand any physiological process in research in biology, first we try to characterize it in using in vitro experiments. That means using some cells, using some cell lines. And when we get any positive result from our in vitro experiments, then we shift it to in vivo experiments. And after that, we can go to human samples and we can do it in human experiments. But the first thing that we have to do is in vitro experiments. And many disease that occurs in human body, if those diseases are also occurring in some kind of animal models, then we can use an animal model system to study about that disease. For example, this gene palbardism, this is a syndrome which occurs in the mutation of the KIT gene, and KIT gene is expressed, basically it is required for some kinds of pigmentations. So it is pigment forming cells which are like ear, or gut neurons, many other genes. So that is it is. So now we'll move to the next question. Okay. The pre-formation theory is supported by the view that a human carries a miniature version of the progeny in the gametes as a homunculus.
Sorry
And
to
this
sorry to interrupt, but correct answer for question one is all or only A and C or A and B or A, B and D.
okay, so I think
Correct answer for last question.
I have already explained. So I'll just request you, Ajay, that please try to follow, because I think I have I have shown this slide. So the question was, KIT gene is expressed in which of the following cells? As you could see from this particular figure, it is expressed in neural crust cells, precursor of blood cells, and primordial germ cells. Three. Now you look at the options. Pigment forming cells, yes. Precursor of blood cells, yes. Germ cells, yes. Cardiac muscles, no. So any of the answer would be correct. If you are choosing A, B, or C, any of them are correct. Cardiac muscles, no. KIT genes is not expressed in the cardiac muscles. Okay. Now, next question. Okay, so the pre-formation theory is supported by the view that a human carries a miniature version of the progeny in the gametes as a homunculus. And this simply grows up to become an adult. Based on your understanding of the theory, which of the following can disprove pre-formation theory? Dalton's atomic theory, variations, presence of a homunculus within a gamete, Malfighi's observation of blood vessels in an unincubated egg, the presence of intermediate phenotypes. This is the question. After 2 minutes, I'll switch the screen. Okay. Okay, I'm switching the screen. Shall I switch the screen? Okay, I'll just switch it. I cannot even see what you're saying. Okay, fine. Let us see what you have answered. Okay. short limb, mostly if we see, like if you have read, so one thing, okay, just a minute. I'm sorry for the interruption. Yeah, okay. Okay, so the answers, what people have answered, C, B, except C, all are correct. Okay, let me see the. Okay. Which of the following can disprove preformation theory? Dalton's atomic theory, variations, presence of a home nucleus within a gamete, Malfighi's observation of blood vessels, the presence of intermediate phenotype. So the correct answers are Dalton's atomic theory, variations, and the presence of intermediate phenotypes. That means, okay, so how many of you have answered correctly? You can see C and B. Okay, I think most of the students have answered correctly only, but I can see very less participation. So everybody please try to answer the questions. Okay. And now first we'll see what is preformation theory, and then I'll explain the question to answer to you. Okay. So I'll explain with the help of the PDF. So it is easier for me also, and it is easier for you also. Okay, I think it is in second class. Hmm. So basically, when the different theories about how an organism develops and how an organism comes. So when different theories were developing, at that time there was a theory which is known as pre-formation theory. So what does pre-formation theory says? That a miniature version of the adult form of our body. So just like how we look. So an adult body looks, whatever the way that an adult body looks, a miniature version of our adult body is present in the germ cell itself. So there is no development as per se. That miniature version gets converted, or I mean it gets developed into the whole organism. There is no starting development of each and every cell, or making of a finger or a limb from initial point. The miniature version of our fingers, our limbs, our head, everything is already present within the germ cell, and it just grows to become a full body. That is known as preformation. That means the miniature version of our body is preformed in the germ cells. Okay, and this preformed in the germ cell, this and so earlier they were, this theory also uh, used to go well with the scientific, with the religious notions of the people of that time. And now another question was that if we are saying that the pre miniature version is present in the germ cell, then now it is present whether in a sperm or an ovum, because both are germ cells. So because because we have a patriarchal society, so it was assumed that okay, the miniature version would be present in the sperm. So they thought that a miniature version is present in the sperm germ cell. So this is a basic idea of the pre-formation theory that they think is the reason of how an organism develops. Okay, now we come to the question. Okay, based on your understanding, which of the following can disprove pre-formation theory? Okay, so Dalton's atomic theory. So what is Dalton's atomic theory? Can anybody tell me? What is Dalton's atomic theory? What does Dalton's atomic theory tells us? So basically, just a general concept of what is Dalton's atomic theory. So Dalton's atomic theory has several postulates that all the elements are made up of small particles which we call it as atoms, and all the atoms are identical, and the atoms cannot be created and it cannot be destroyed also. Okay, so this is what Dalton's atomic theory essentially deals with. You must have studied about Dalton's atomic theory in your chemistry also. So now they are saying that at all the uh, uh, elements are made up of atoms, and atoms of one element are completely similar to each other, and atoms have protons, electrons, neutrons. So this is just the chemistry aspect of Dalton's atomic theory. That means that individual molecules, for to make an element which has more than two molecules, the individual molecules, they come together and assemble to make an element. But they are saying that the organism is preformed. That means everything is already made, so there is no need of individual molecules to come together to make an element. So Dalton's atomic theory can disapprove the preformation theory. Then variations. Now what is the meaning of variations? Variation means that, for example, a child will not be a complete look alike of his parents. Okay, there will be differences in the body, in the facial expressions, in the physical capabilities, in the height, in color, everything. We are not completely clone of our parents. Okay, there are variations, and apart from these variations, there are certain variations that an organism develops over a time because of environmental conditions as well. Okay, so those are known as epigenetic variations, epigenetic modifications that occurs throughout the lifespan. So if a miniature version of our own self is present inside the body, then how the variations are arriving? So variations can disapprove the preformation theory, because variations are a basic building block for evolution. Evolution has happened, and evolution will happen because an organism accumulates variations in the form of environmental epigenetic changes, in the form of mutations, and in the form of how an organism is genetically made. We know that there are certain genes which are dominant, certain genes which are recessive. So it's not like the dominant gene will always express. If you know a little bit of genetics, sometimes if both the parents have that recessive allele, that recessive gene can also express. Although, for example, for a coat color, suppose orange coat color is a dominant coat color and yellow coat color is a recessive coat color. Now both the parents have an orange coat color because they have that orange coat color dominant gene, but they also have yellow recessive gene. So that recessive genes, both the recessive genes are coming together, that their offspring will have a yellow coat color, although both the parents are not yellow coat color. So that means there are variations. The offspring is not the carbon copy of the parents. So variations are there. Then the presence of intermediate phenotypes. Just this, the presence of intermediate phenotypes. That means blue and yellow and sorry, white and red can give a pink colored flower also. It will not always be that if a white color flower and a red color flower is there, so only red color will always come. A pink color flower can also come. If you have studied about epistasis, then you can relate with it. So these theories can disapprove the preformation theory. Okay, now presence of a homonucleus within a gamete, no, it doesn't disapprove the preformation theory. Why? Because we are just saying that homonucleus that we stated, it is present in a sperm, it is present in a gamete. So it is same thing, you're just changing the germ cell. Then Malfighi's observation of blood vessels in an unincubated cell, no, it was the first time observation. If you can see here, it is given here also. In the first or second slide, I think. Sorry, second slide. Yeah. Yeah. See, the Malfighi's observation also stated the preformation theory also. It also emphasized on the preformation theory only. So this is again is not disapproving the preformation theory. Okay. Next question is, Phocomelia is a rare congenital anomaly which is associated with loss of external ear and caused by thalidomide, severe shortening of the limbs and caused by thalidomide, loss of vision and caused by a mutation in the KIT gene, and severe enlargement of limbs and caused by mutation in the KIT gene. Okay, so I'll give 2 minutes. You can go through the question, and after that, you can reply in the message box. And I will try more and more students should participate. It doesn't really matter whether you are giving a correct answer or not. What matters is that you are trying. Okay, because I can understand that you are doing your bachelor's or masters, you have your classes as well. So, coping up with the classes, this NPTEL lecture can be a little tiring and cumbersome, but since you have opted for the course, you have to go through the lectures. So, even if you haven't gone through the lectures, at least try to answer it. Okay, in 1 minute, I'll switch the screen.
Samra,
Sorry, you have attempted. If you have attempted, that's fine. Just wait for a minute. Okay, I think I'll just switch the screen now. Okay, good. I think many people. Just a minute. Okay, so I'll just small request, try to be on time. I understand that you have your classes and everything, and your schedule might be very busy, but you can be 5 minutes late, but it's 6:38, almost more than half an hour. So try to be on time. Okay, so let's see what you have answered. Shivani, so whatever extra questions you have, I'll answer it after we are done with the questions. Okay, we'll have ample amount of time. First, we'll try to complete what we have to do for the syllabus, and then we'll see the other questions. Okay, I hope you can wait up till we finish the questions. Okay, so most of the students have answered B. Okay, there is one A also. So I'm sorry if I pronounce your name incorrectly. Rajshri, Sudhar, Devnath, Bhavna, Ajay, and Shivani have answered as B. And Namaninda has answered Shivam. Okay, I'll say Shivam. That's fine. That's better. Shivam has answered A. So let me see what were the options. Okay. So the correct answer is B. Okay. The correct answer is B. That is severe shortening of the limbs and caused by thalidomide. Okay. Now let us see here. Third lecture. Yeah. C. Can you see here? Developmental anomalies caused by environmental agent thalidomide. So you can see here that this particular drug, it leads to severe abnormalities and shortness of the limbs. Okay. Hmm. Now, very interesting thing about this is that pregnant ladies, if they get exposed to this drug, then their, their offspring or their child would have this disease. And but if what kind of disease, basically it would vary about according to the days of the menstrual cycle of when you are taking this drug. So mostly if you are in your uh, conceiving phase and you take this drug, then the most important or the most severe form is the shortening of the legs for the limbs. But if you are taking this medicine apart from I mean when you're not pregnant, then this thalidomide has certain advantages also. So can you tell anything about this drug thalidomide? Anything that you know about this drug? For which conditions basically it is used?
This drug was used in the pregnancy in nearly 400 patients
Hmm.
to avoid the
To avoid the.
morning sickness. Means the
Okay.
pregnant lady gets
Hmm.
vomiting and
Good.
that is called as.
Yes. And what
But
happened
later
then
on,
after that? What
this
was observed?
because of this drug, there are 400 patients got this, and this is the data about these patients.
Hmm.
Even A would have been correct if you had been mentioned anoxia.
Hmm.
Phocomelia especially refers to the shortening of the limb. That is a condition for the limb,
Yes.
phocomelia, due to thalidomide. Now, this drug is again because it is acting on DNA, so this drug might be coming to be used in some cancers.
Hmm. Yes. So now this drug is used for the treatment of some kind of myelomas or some skin disorders and some other kinds of cancerous conditions. The only precaution is that this drug cannot be taken by pregnant ladies. Pregnant ladies should not take this drug. So this drug has advantages also, but just that like many things, like a pregnant lady must should avoid many drugs and many things. So along with them, this thalidomide cannot be taken by pregnant ladies, but this drug is still used because it has applications in treating severe kinds of tumors and cancers. Okay, good. Now, next question is, a researcher wants to study the development of melanocytes. Which of the germ layers would yield the cells of interest? It's a simple question. Ectoderm, mesenchyme, mesoderm, endomesoderm, and endoderm. I'll give you 1 minute, 2 minutes, and then I'll switch the screen. Okay, 1 more minute. Okay, fine. I'll switch the screen now. Let us see what you have answered. Okay, just let me see the options first. Um, melanocytes. Okay, fine. Okay, sorry. Hmm. Okay, I think most of the students have answered it correctly only. So, Sudhar, Bhavna, Rajshri, Anand, Devnath, Shivani, Ajay, Shivam. Okay, that those are the previous ones. So I think everybody's has. Okay, I will. Okay, sorry, I'll say you. Camellia. I don't know whether I'm pronouncing it correct or not. Can you tell me your pronunciation, please, by unmuting yourself? Am I pronouncing it correct? Okay, fine. Okay, so Camellia has also answered. I think everybody has answered it correctly only. The answer is A, that is ectoderm. Okay, so we all know that we have three extraembryonic layers. The first one is the ectoderm, then there is mesoderm, and then there is endoderm. Okay, so these three layers are formed at which stage of the development, during development? Can anybody tell me at which state for the first time we see these three layers? Anybody?
Blastula to morula stage.
Yes.
First the blastomeres form, and after that the morula stage is. At that time we see these three layers.
Yes. So right after fertilization, what happens? Your cleavage starts appearing. Okay. And then your one cell is divided into multiple smaller compartments. Okay. And these smaller compartments actually partition the cytoplasm into smaller cells. And those smaller cells are known as blastomere. And your at the end of the cleavage, what is formed is blastula. So right after your fertilization, what is formed? Blastula is formed. Now, what happens that after the blastula is formed, your blastomeres, which are the smaller cells within the blastula, those blastomeres migrate and rearrange into three layers. And those three layers are ectoderm, endoderm, and mesoderm. And then it forms a gastrula. Okay, so so the correct answer here is ectoderm. Yes. So from blastula, gastrula is formed. So blastula means after cleavage, your diploid cell, it divides into multiple smaller compartments, and each smaller compartment, the new smaller cells which are formed, those are known as blastomeres. They are known as blastomeres. And then what happens that these blastomeres, they migrate and rearrange to form the three layers, which are ectoderm, endoderm, and mesoderm. Okay, so these three layers are formed, and these three layers will give rise to everything. Now, ectoderm means outer, endo means interior, meso means middle. So what are the organs that comes under ectoderm? The surface layer of the skin, your brain, your nerve cells, those comes under ectoderm. Your endoderm, what would be like your lungs, your intestine, that all comes under endoderm. Your mesoderm means your blood, your heart, your some facial muscles, your kidney, those all comes under endoderm. Now the question here is, if you see, the question is saying the researcher wants to study the development of melanocytes. Now what are melanocytes? Can anybody tell me what are melanocytes?
Color pigments of the skin.
Big pigment forming cells. So
Pigment coloring the skin.
Melanin.
I don't know what just happened. Anyways, so melanocytes, what are, I was asking what are melanocytes? I couldn't understand what you said. There is some, I mean, your voice is not that clear, it seems. So I want anybody else to try. Ajay, if you know the answer, you can just I want others also to participate in the class. So anybody else who wants to tell what is melanocytes? Anybody else?
Producing color pigment.
Yes. So basically, melanocytes are the cells that produce the pigment which is known as melanin pigment. Okay, and this pigment, pigment, gives color the gives skin the color. So if someone is very fair skin, that means this pigment is less in them. And if someone has a little darker complexion, that means this melanin pigment is present more in them. Okay, so this pigment is responsible for the color, and this pigment also kind of protects us from a lot of UV radiations and from a lot of skin cancers as well. So now we are talking about melanocytes. Melanocytes are present in the surface layer of the skin. That means which part we have to target? We have to target the ectoderm. Okay, so this is the answer. I was just explaining. So many students already know the answer. So but my thing is that I will try to explain in a way that even uh, everybody can understand. Okay, some of you are some of you must have studied these things. Some might not have studied these things, and some are studying it for the very first time. So that's why I'm taking from the basics so that everybody can understand. Okay, this we have done. Okay, yeah, this question. What is the question? Okay. If you have to create a fate map only by direct observation of the developing embryo, which of the following features would you choose? Organism having a large number of cells, organism having many non-pigmented cells, organism having large sized cells, or organism having a small number of differentially pigmented cells. I'm giving you 2 minutes. You think and then we'll see the answer. Think this question carefully. Even if you do not know the answer, it's fine. Just try to attempt something. We'll study detail about what is fate map. Everything we'll study right now. Okay? Okay, so should I switch the screen? Okay, I'll just wait for 1 more minute and then I'll switch. Okay, now I'm switching the screen. Okay. So Sudhar has answered D. Nabandita has answered D. Rajshri has D. Anand D. Camelia D. Shivani D. Ananya D. Ajay D. So it's good that everybody has answered it correctly. Okay. So the question is, if you have to create a fate map only by direct observation of the developing embryo, which of the following features would you use? Okay. So even if you know nothing about what is fate map and what is you know nothing about this question, but even with the presence of mind, you can at least try to solve this question. How? Because direct observation means that just suppose if you have to differentiate between two things, so would it be easier for you to differentiate if they are of different colors, or easier when they both are of same color? If I have an object, if I have something as A which is also red color and B also red color, then is it easier to differentiate when they both are of same color, or when both are one of them are of different colors. If A is of red color, it is of green color, then it is easier to differentiate now. So that means pigment means color. So if any organism which has different pigmented cells, it is easier to observe it directly. I mean, for direct observation, it is easier to observe cells which are of different pigments. One thing. Second thing, just randomly think that if you have to directly observe, then if you have small number of cells, then it is easier. You can count the cells, you can carefully monitor all the cells. Or if you have large number of cells, it would be a little difficult. So if an organism has a smaller number of cells and differentially pigmented cells, that means the cells are of different colors, then it is easier for it to observe directly. Okay, so just like a general logic I am telling you now. So the correct answer is D. Now let us see what is fate mapping. Okay, so fate mapping is basically a technique to trace cell lineage in developmental biology. Okay, so basically, if you have an embryo and you want to see how that embryo, how the cells are dividing, how the cells are migrating during developmental process, how the cells are differentiating to become more mature and highly advanced cells, that means you are trying to trace their cell lineage. So fate mapping is nothing but tracing of the cell lineage. Now this fate mapping can be performed by various techniques. The first one is direct observation. The second one is vital dyes, fluorescent labeling, radio labeling, and genetic mapping. Okay. So direct observation means that directly you just by the morphology, you are trying to get some idea about the fate mapping. Vital dye means that there are certain dyes that do not harm the organism. Okay. So you cannot take any dye for fate mapping, because dye only those dyes can be taken which are not harmful or detrimental for the life of the organism. So certain dyes which does not harm the cell division or which does not harm the development of the organism, those dyes can be used to incorporate into the cells, and then as the cell divides, that dye would be there within the cell and you can trace it. Could you understand? Like if you are giving that dye to the embryonic cells, and as the cells will divide, the dye is also there with the cells. So the dye will also eventually go with the cells and you can try to trace it. Just like marking something. Okay. So this is what vital dye can do. But there was a problem with vital dye. What is the problem? That with every round of replication or division, the vital dye gets diluted. So it is not very easier, it is a little complicated when we use vital dye. It is obviously it's better than direct observation, because the dye will get diluted with each round of division or replication. So then what we can use fluorescent labeling. Fluorescent labeling means that we can use some fluorescent molecules, and those fluorescent molecules, when they are attached, then we can trace them, because fluorescent molecules will not get diluted, because fluorescence is a property. Okay, then we can also label ourselves. Radio labeling can also be done. And then the most advanced and highly used now process, which is the genetic mapping. Genetically, when we are trying to uh, trace the lineage of a cell. So this is about fate mapping. We can also see it in the mm, PDF if you want to. Where it is? Yes. So you can see direct observation. There is one technique. Second is use of vital dyes. So, mm, yeah, use of vital dyes. So, when we are using vital dye, we are actually staining the cells. So, when we are staining with different colors dye, you can see the different parts of the organism. And then we can use fluorescent labeling and we can use radio labeling. These are just the examples you can go through. And then also we can use the genetic genetic way or genetic mapping, which is the most recent and advanced version which people usually do nowadays. Okay, uh, fine. Okay, someone wants to join. So, Shubha or Subha, so can you tell me why you joined so late? It's 1 hour is almost is over, completely over. So we have already discussed some questions. So it's my humble request. I understand that you might have your classes and the schedule is busy. But if I am here, if I come and I join it, I'm 10 or 10, 15 minutes before only I come for the class. And I'm doing PhD. So my stress level is different only. So if I can take time for you guys to come, then it would be better if you can also come on time. 5 10 minutes is okay, but 1 hour is little too much. Anyways, try to be on time for the next time. Now we'll do the next question. Which of the following is or are present in all vertebrates? Fertilization, cleavage, organogenesis, metamorphosis, viviparity. I'll give you 2 minutes. You think, answer, and then I'll switch the screen. I think it's easy one only. Okay, now I'll switch the screen. Let us see what you have answered. Okay. Oh God, people are now coming. Fine. Okay. So Anand has answered A, B, C, D. Let me see the options first. A, B, C, D. Okay. Rajshri has answered A, B, C. Sudhar has answered A, B, C. Camelia has answered A, B, C. Navanita has answered A, B, C. A, B, C. Anand, can you give one example of vital dye? So vital dye, like acridine orange or methylene blue, or there is one type BBG. I am not BBG stands for, I think, brilliant blue, something you can see. So bromophenol blue, acridine dye, acridine orange. These are the examples of, or you can have fluorescein also. Fluorescein is also an example of vital dye. So there are many. Okay. Then, okay, fine. So most of them have answered it correctly. Anand, can you tell me why you have answered, why you have opted A, B, C, and D? Do you think metamorphosis occurs in each and every vertebrate? So tell me one thing: are humans vertebrates or invertebrates? Humans are vertebrates or invertebrates? Anand, can you tell? Humans are vertebrates or invertebrates? Vertebrates. So, do does humans have metamorphosis? Do we metamorphosize? So that means metamorphosis is not present in all the vertebrates. Okay. So here they are asking which of the following are present in all the vertebrates. So fertilization, every vertebrate occurs fertilization because when egg and when sperm and ovum it combines, fertilization takes place, and after fertilization cleavage takes place, and after cleavage your blastomere is formed, gastrula is formed, and then your eventually your organogenesis, that means formation of organs, takes place. But metamorphosis occurs in each and every vertebrate? No. Viviparity, does viviparity occurs in each and every vertebrate? No. Now, can you tell me what is viviparity? What do you mean by viviparity? Anybody? Viviparity means what?
From development of embryo inside the body.
Okay. So, according to this, humans are what? Viviparous? Yes or no? Humans are viviparous or
Yes,
not?
ma'am.
Yes. So humans are viviparous because fertilization and embryo development both is occurring inside the body. Okay. And uh, then what is oviparous? What is oviparity? Can anybody tell me what is oviparity?
Production of eggs inside the body.
Yes. So
Egg is
basically
produced outside the
outside.
body and rest of the development occurs in that egg. That is oviparity.
That means animals, they are laying fertilized eggs outside the body for, and then the egg is developing outside the body. For example, if you see birds, they lay eggs and then they sit on it for it incubation. So that sitting, when you if you have observed that birds, they sit on their eggs. So what they are doing, they are actually providing by the warmth or that heat and that incubation time for that egg that can hatch and become a complete organism. So that is your oviparity. And what is oviviparity? Oviviparous. What
Is
is?
as soon as egg is laid down, the full developed organism is in. Like I think in snakes or in marshes.
Mm, uh, can you repeat it, Ajay? Sorry, I think there's some network problem, so I'm unable to hear what you're saying.
As soon as egg is laid down, the whole an organism is developed inside. It just
Hmm.
hatches out and come out of that
Hmm.
egg.
Okay.
Means
For example,
mix of
can you give any examples like snakes and?
dinosaurs and marsupials, things like that.
Yes.
They are mammals, but they lay eggs,
Hmm.
and organism is developed inside.
Okay. So I think the question would be, I think, clear that fertilization, cleavage, and organogenesis, A, B, and C. These are the three things which are present in all the vertebrates. Okay. Now, next question is, yeah, with respect to fate mapping, what are the advantages of creating a genetically labeled organism over the use of vital dyes? It labels cell permanently. It doesn't get diluted out with increasing number of cell divisions. The genetic label can be easily identified, or all of the other options. 2 minutes to answer the question and then we'll switch. Okay. Okay, now I'm switching the screen. Okay, let me see which with respect to fate my how. Okay. Let us see what you have answered. Okay. So that has answered C, Anand C, Bhavna D, Navandita D. Okay. Camelia all, Ajay D. Okay, fine. So one thing just now as we just studied that vital dyes it gets diluted by increasing number of cell divisions. Okay, so vital dyes will get diluted by increasing number of cell divisions. But when you are genetically labeling an organism, that label will be there up to n number of cell divisions. That means as many cell divisions as is occurring, that genetic label will be there with the cell. Okay, so this is one of the phenomena which is observed when you are labeling an organism with a genetically labeled dye. Okay, I mean genetically labeling. So that means it will not get diluted. Okay, because it is not getting diluted with increasing number of cell divisions, that means what? It is labeling the cell permanently. Yes, and because it is labeling the cell permanently and it is not getting diluted with increasing number of cell divisions, that means the genetic label can be identified easily. That means what would be the correct option? All of the options. That means genetically labeling an organism is better than using vital dyes because this genetic label will not get diluted with the increasing number of cell divisions. And because it is not getting diluted with increasing number of cell divisions, that means it is labeling the cell permanently. And because it is labeling the cell permanently, it can be easily identified. So the correct answer is all of the other options. Okay. So Anand and Sudhar has opted C. And C was the genetic label can be easy. Okay. Fine. D is the correct option. I think now you must have understood why. Okay. Next question is the uh, the mouth of Xenopus laevis is referred to as 2 minutes. After 1 more minute, I will switch the screen. Okay. Okay. Half a minute more. I'm switching the screen now. Sudha has answered E. Bhavna has answered E. Namrata has answered E. Kimberly has answered E. Rajshri has also answered E. Okay. Ajay has answered E. Okay. What is E option? Yeah, stomodium. So I think all of you have answered it correctly only. So the question is, the mouth of Xenopus flavis is referred to as archenteron, blastopore, pharyngeal arch, stomites, or stomodium. So the correct answer is stomodium. Now, now, quickly, can anybody tell me what is the meaning of stomodium? What is stomodium? Anybody? So all these A, B, C, D, E options that are here, these all options are actually referring to some of the, some of the preliminary mm, formations in the embryo that leads to the formation of the organ of that particular organ. So if I talk about stomodium, so basically it is present in the ectoderm, okay? And this is the region that develops into mouth and oral cavity. So there is a depression in the ectoderm of a young embryo, and that depression it confines to the oral region that will develop eventually into mouth and oral cavity. Okay, so these all are actually the formations which are observed in the embryonic stages, which leads to the development of respective organs. For example, if we talk about stomodium, stomodium will develop eventually into mouth and oral cavity. Okay, then we talk about gastrocele, and gastrocele is also known as archenteron, and it is basically a cavity. Okay, and this cavity um, kind of um, eventually it will decide uh, the region uh, from head till bottom. That means it will decide and forms the animal's mouth and animal's anus. Okay, then if we talk about somites, what are somites? Basically, somites is like a segments. Okay, somites are basically segments of a particular organism. So a body is divided into many segments, which are mostly longitudinal segments, and those segments are known as somites. And then we talk about pharyngeal arc. What is a pharyngeal arc? Basically, pharyngeal arc will eventually develop into your head and neck. Okay, so these are the different formations that are found in the embryonic stages that leads to the development of either uh, mouth or head or neck, or the mouse to anus region, or the segments of the body. Fine. Okay, now we'll go to the next question. A group of mammals returned to water during evolution to avoid. Okay, I'll just do it in big screen. Fine. A group of mammals returned to water during evolution to avoid competition on land and utilize sea resources. To suit their need in the new habitat, they evolved the ability to swim. Okay. Meanwhile, the avian class evolved the ability of flight to suit their needs. Fine. These are the examples of adaptive radiation, analogous structures, descent from a common ancestor, homologous structure, या heteropoda vertebra. I give you 3 minutes for this question. Okay? See the question, think carefully, and then try to answer. Okay? 2 more minutes. Think carefully what they are asking. Okay. Now I'm switching the screen. Let's see what you have answered. Okay. So Namanita has answered A, C, and D. Okay, okay. Still you're answering? Okay, fine. I'll just wait for 1 more minute then. Okay, fine. Sudhar has answered A, Navnita has answered A, C, and D. What are A, C, D, A, C? A is adaptive radiation, C is descent from a common ancestor, and D is homologous structures. Okay. Rajshri has answered A, adaptive radiation. Okay. Ajay has answered C and D. Okay. C and D means descent from a homology. Okay. Camellia has answered B. B means analogous structures. And Bhavna, Sudhar. Okay, 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 okay. I think those are different questions. Okay, fine. Okay. So mostly people have answered A, B, and C. No, B. Oh God. Sudhar, you are saying okay. A and B. Okay, A and B you are saying. Fine. The correct answer is descent from a common ancestor or homologous structures. Now, a group of mammals returned to water during evolution to avoid competition on land and utilize sea resources. To suit their new habitat, they evolved the ability to swim. Meanwhile, the avian class evolved the ability of flight to suit their needs. What are these examples of? So that means that initially both of them were present on land. Okay, but since there was a lot of competition on land, so what they thought that some of them thought that they'll go to the sea and utilize the sea resources. Okay, and some have thought that they will go to I mean they will, if they have evolved himself in such a way that they have the ability to fly. That means initially they were the same, but they have evolved from the same ancestry, and now both of them are performing different functions. Okay, that means they have descent of a common ancestor, but now they have evolved in such a way that now they perform different functions. Okay, so these structures which are coming from a common ancestry, from a common origin, and they perform different functions, they are known as homologous structures, and this is known as homology, that is descendant from a common ancestor. And what is analogous structures? That they perform similar functions, but they are not descendant of a common ancestor. Whenever we talk about common ancestry, it is always homologous. Okay, so can anybody tell me that what is the difference between adaptive radiation and homology? Anybody? Try. Anybody who knows the answer? See, uh, basically, when we talk about adaptive radiation and homology, both are the concepts that helps us to study the evolutionary process only. Okay, so in adaptive radiation also, can you give me okay, first of all, can you tell me what is adaptive radiation? Anybody who wants to try what is adaptive radiation? I think this way it would be easier for you to understand. What is the meaning of adaptive radiation? Anybody who wants to try? What is adaptive radiation? Do you uh, know about Darwin's finches? The Darwin's finches?
Um, yeah, ancestor finch species, diverse multiple
Sorry, Sudhar, what are you saying?
ancestral finch species diversified into multiple species.
Yes. So
Darwin species.
Darwin's finches is an example of adaptive radiation. So both of them are spreading and trying to evolve according to their environment. In adaptive radiation, also, what happens is that the they try and they try to uh, they try to adjust themselves according to the environmental conditions from one particular geographical area. They spread to different kinds of geographical areas, and they try to adapt themselves according to their different geographical area. For example, you have Darwin's finches example. But when you talk about homologous structures, so homologous structures, whenever you talk about homology, very two important points are there, that they have common ancestry, first of all. Secondly, is that mostly what happens in that homologous structures need not to perform similar functions. They do not perform similar functions. Darwin's finches, they perform similar function only. The way they were performing, that was different. Okay, finches, the finches have evolved according to the geographical conditions, but those were still finches only. They were performing the same function that it was destined for. The way it was performing the function, that has changed. But when we talk about homology or common ancestry, that means the only concept that you have to keep in mind is that they have a common ancestor. After that, when they have radiated, after then when they have diversified, whether they are performing common function or not, we don't know. Whether they are performing similar functions or not, we don't know. But they have a common ancestry. So very important to understand between these two terms, homologous and analogous. So now can anybody explain me what is the difference between homologous structures and analogous structures, or homology or analogy?
And so when
Okay, Ajay, you can speak.
homologous means it is about the common origin, but it may be same function or different function.
Yes.
And analogous means there is a it is just a functional thing is same.
Hmm.
It is not the common origin.
So
Means ancestry is
analogous,
different.
hmm, analogous structure will not have a common ancestry, but the function of the function would be similar. So what we have to understand, that same function does not tell that they have same ancestry, or other way around, same ancestry need not to have similar function. Okay, so ancestry and function, these two are different terms, or they have different relevance. Okay, now we come to the next question.
Madam, sorry to interrupt. There are two terms also I came across, don't know where. They were
Okay,
autologous and orthologous.
okay, yes.
I
So
was
there are two.
autologous I actually understand from the same person, but orthologous I was not able to understand.
Okay, so uh, do you know? Have you heard about the term paralogous also?
Yes, madam. Paralogous means in the different species. having same means at a that unless the in between part is cut, that remains on the over the
Okay.
same part of the bone.
Okay. Now I'll
No.
explain what is orthologous and what is paralogous also, because whenever we study about orthologous, we have to study about paralogous also. Okay, just a minute. I'll have a new slide. So I told just now
Just like gene duplication occurs, they
homology.
are removed during the evaluation.
Hmm. Okay, I'll explain it. Homologous. Homologous means common ancestry. Okay, this is clear. Fine. Now, homologous structures can be subdivided into two types. Okay, one is orthologous and the other one is paralogous. Okay, so both are homologous genes only. That means both have common ancestor. But what is the difference? That one has evolved due to speciation event and other has evolved due to gene duplication event. That is the only difference. So when we talk about paralogous structures, so these are the genes which are present in single organisms only, but they are the genes that have arised due to gene duplication events. Okay, so these are because of gene duplication events, whereas genes which are separated due to speciation events, those genes are known as orthologous genes. So, homologous genes are the genes which have a common ancestry. And in homologous genes, see, common ancestry, ठीक है, but these common ancestry have now diversified themselves. Now, this diversification can be because of gene duplication, or this diversification can be because of a speciation event. If the diversification is because of gene duplication events, then those genes are known as paralogous genes. And if the diversification is because of speciation events, then those genes are known as orthologous genes. Okay, so orthologous and paralogous, it's important to understand. And there are many examples. Currently, I cannot recall an example. You can just Google it. There are many examples you can see. But important point are speciation is orthologous, gene duplication is paralogous. And why this is important, why I'm focusing more on homology and analogous genes and orthologs and paralogs, because these are very very frequently asked questions in your gate examinations or in CSIR examinations. So up till now, whatever we have studied, so what I'm thinking is that whatever lectures that we are doing, whatever slides or whatever content that we are reading, I'll try to highlight the points that are important not only for your NPTEL exam point of view, but also for your competitive examination point of view. Because if you want to go for higher studies, so let's say PhD or masters, then you have to give some competitive examinations, right? So in competitive examinations, especially when you're giving CSIR net, then your developmental biology is a very important topic. So this and in any general competitive examination also, orthologous, paralogous, homologous, analogous, these terms are frequently asked. What is the meaning? So that's why it is important to have a clear idea about what are homologous genes, what are paralogous genes, or homologous structures and analogous structures. Okay. Now we come to the next question. Yeah, flu protein. So flu protein is what? Homologous or analogous? Sorry, flu protein is a orthologous gene.
Autologous.
Yeah, orthologous gene. Fine. Now we'll come to the next question.
Common is hemoglobin in human being.
Okay. So I think this is the last question. After this question, so what was the name? Uh, Someone has asked a question Shivani, can you just paste the question again? The question that you have asked. I actually now it's very difficult to do again. Please paste the question again. Meanwhile, we'll solve the question, the last question for the day, and then we'll come to your question. You have asked one question, I guess, and I told you that we'll answer. I try to answer the question at the end of when we complete all the questions. So if you can just paste the question again, so that we can discuss on it. Okay, fine. Okay, I'll give you 3 minutes because it's a matching. So match the following. I'm not saying anything. You have to just see the process and the structures: delamination, matrix secretion and degradation, cellular intercalation, and cell death. There's no much concept to explain. You just have to memorize it. But even then, I'll tell you where it is in the PDF. It's actually very easy, even if you do not know the answers, even if you know just one of them, I think you will be able to solve it. It's not that difficult, actually. One more minute. Okay. Shall I switch the screen? Has everybody tried to answer the question? Okay, I'll switch the screen now. Okay, let us see. Mm, okay, only two students have answered. Why? Others also try. Okay, so Sudhar has answered. It is second one. Okay. And Ajay, A ka G. A G, okay. B H, sorry. Matrix secretion. B H, okay. C E. C E and D F. D F. This is an option. A G B H. Okay. Others also try. If you have gone through the lectures, I think you would be able to answer it because it's easy actually. It's just memorizing. There's nothing much to explain. Amelia, what do you think would be the answer? Or Bhavna, Subha, Navandita? I'm sure I'm pronouncing your name incorrectly. I'm sorry. Okay, fine. It's okay. See, even if you do not know the answer, one thing which is very important while you are answering multiple choice questions is your presence of mind. Okay, and you should try to think logically, even if you do not have very much conceptual knowledge about the topic. Because if you try to think logically and you will try to use your mind, then it will help you for your competitive examinations also. Okay, so here, any if even if you know anybody, like any of the options, for example, let's take cell death, interdigital mesenchymal. So what happens is, uh, that some organisms, they do not have distinct fingers. They have a web. For example, bat. Okay, so they have a web. So that means that their fingers are not distinct. That means the cells which are between two fingers, they are still present. That's why their hand looks like a web and not distinguished fingers. But when we talk about humans, we have a web or we have a hand? We have a hand with distinct fingers, right? We do not have cells between our fingers. Do we have? No. That means the cells which were between the two fingers, which actually help to form a web, those cells, our body has evolved in such a manner that those cells have died. That means those cells have been programmably killed, which is known as program cell death or apoptosis. That means in our body, program cell death occurred, and because of that program cell death, the cells which were between the fingers have died, and that is because we have different distinct digits, different fingers. So that means cell death would be related to interdigital mesenchymal. Mesenchymal is a cell type. So we have epithelial cells and mesenchymal cells. We'll discuss that later. Okay. Second is delamination. What is the meaning of delamination? Can anybody tell me who have answered correctly? Ajay, you have answered it correctly. So can you tell me what is the meaning of delamination? What do you think? What is the meaning of delamination? See, delaminate. So what in general, what is the meaning of laminate? When we laminate something, what we do? We put a thin layer of sheet over it.
Something which goes inside. Actually, it occurs in west. Sorry,
Hmm.
I interrupted. Invagination during vestibular stage, it is called as delamination. Something goes inside.
Hmm. Yes, that means, so in a very simpler term, I'm just trying to relate that lamination means, for example, you have a sheet and you put a thin film over it and you laminate it. Okay, so you're putting a film, you're putting a sheet over it, over it, and delaminate means you are removing that sheet. So during the process of gastrulation, what happens is that your endoderm actually kinds of is divided into different thin sheets, not sheets, but thin layer of thin cells layer. Okay, and that is known as delamination. That means your endoderm is split into different layers, and it kinds of invaginates and it forms different layers. That is known as delamination. Okay, so
Actually,
other
it is a
concern.
process to change it to triploblastic layer. Otherwise, there are some example. They are having diploblastic layers. We are human being, and all vertebrate they are having diploblastic.
Hmm.
No
Okay.
delamination, then only it will be endoderm and ectoderm. There will be no mesoderm.
Hmm.
That's my understanding, madam. Sorry.
Okay. I think rest of the terms, matrix secretion and degradation and cellular intercalation, are Simple only. There's nothing much to explain. So matrix secretion and degradation means, so B should go with huh, mesenchymal cells. Okay. So one thing which is important for you to understand is, okay, so some students are still joining. Mm, mm, yes, fine. So there are two kinds of cells: epithelial cells and mesenchymal cells. So any kind of terminologies like matrix secretion or matrix degradation comes, then mostly they are related to mesenchymal cells only. Why? Because the property of mesenchymal cells are that they are what kind of cells? Can anybody tell me what is the difference between an epithelial cell and a mesenchymal cell?
Epithelial cells are attached to each other, whereas mesenchymal cells, they are free.
Hmm. So basically, it's a loose tissue. I mean, it's a loose connective tissue. So that means epithelial cells will be attached, and they are um, uh, cuboidal in shape also. And mesenchymal cells are like spindle-shaped cells, which are loose cells, which can move and migrate. So whenever we talk about degradation, or when we talk about matrix secretion, then the cells which are generally involved are mesenchymal cells. Also, mesenchymal cells are very important not only during the embryo development, but mesenchymal cells are also very important um, during when the uterus is preparing itself for the incoming blastocyst. The incoming blastocyst will come and sit on the uterus. So uterus layer, it kinds of prepares itself for the incoming blastocyst. So at that time there is a process which is known as epithelial to mesenchymal transition, EMT. So this EMT process occurs, and this what it does is that the uterus kind of remodels itself, and many of the epithelial cells, they actually try to convert to mesenchymal cells, and in doing so they are actually preparing themselves for the incoming blastocyst. So this EMT is very important phenomena. And also interestingly, this EMT, epithelial to mesenchymal transition, is also observed in many kind of cancerous conditions. So in many kind of tumor conditions also this EMT phenomena is observed, because this uh, mesenchymal cells, these are these are loosely packed cells. They can easily move, they can easily migrate. So when they can easily migrate and they can easily move, they help in metastasis. So that is why this epithelial to mesenchymal transition is observed in cancerous cells. Cancerous cells metastasize, that the cells from their primary position, they go and try to spread to the entire body. So in that spreading, the mesenchymal cells helps them. Okay, so now we'll try to look it in the PDF where this topic is there. mm, yeah, here it is. You can see migration means cells move at particular times and places, and the example of migration is heart mesenchyme. Matrix secretion and degradation means synthesis and or removal of the extracellular layer. That means the extracellular layer, it is synthesizing itself, and some kind of old layers are removing from there. Degradation is happening, and the example is cartilage mesenchyme. So then delamination means epithelial becomes mesenchyme part of structure. This occurs in chick hypoblast. And cell migration, which is intercalation, rows of epithelia merge to form fewer rows. You can see here there are two rows here, but they have merged in such a way that it has formed one row, which is observed in gastrulation. Matrix secretion and degradation means synthesis or removal of the ECM, that is extracellular matrix. So why I am saying you should why I just pointed out this table is that because for this assignment, if some questions have arrived from this table, then for your next assignment, or if not for an assignment, for your exams, some other questions can arise from this table. So try to just go through it and try to memorize the examples and try to understand the concept behind it. So this table is important because one matching has come. So you can just go through this table and try to memorize it. Okay, now we'll try to answer that question that one of the student has asked, but she hasn't replied me. So I have to go to the chat to find. Uh, Shivani, ma'am, I didn't get my answer. Waiting for your response. Okay, let us see what was her question. This was her question. So her question was, ma'am, short limb is also associated with mutation in the KITG. Okay. So we have studied that KIT gene is responsible for which of the condition? Some skin related condition, right? Uh, it was in the third lecture only. Yeah. See, so KIT gene is responsible for some skin condition which is known as peel baldism, where you observe a patch. So the answer, according to my understanding and my knowledge, it is no. That short limb is not associated with mutation in the KIT gene. So KIT gene is actually an oncogenic gene. If you just study about KIT gene, so KIT gene is responsible in many kind of skin related diseases, some kind of skin cancers, and some kind of other kinds of cancerous conditions. So it is basically not an oncogene, I can say. It's a proto-oncogene. So, so I don't think that it is involved in this thing, short limb. So can anybody tell me the difference between an oncogene and a proto-oncogene? Anybody? What is the difference between an oncogene and a proto-oncogene? So basically, there are some genes, for example, just like a general way, that in tumor conditions, in cancerous conditions, what happens? Cells continuously proliferate. Okay, there is uncontrolled proliferation of cells, which leads to the accumulation of tumor mass. That is your tumor. Uncontrolled proliferation of cells. So now the normal proliferation is not happening. Proliferation is uncontrolled. Now this proliferation which happens, that is also under control of some gene or not. Some gene must be responsible for the proliferation. So yes, there are certain genes in our body which are responsible for maintaining the proliferative capacity of cells. And if those genes gets mutated, then what will happen? If there is a mutation in those genes in such a way that your proliferation is happening uncontrollably. So if there is a gene which is responsible for proliferation, and now that gene has got mutated, and now the mutation is causing uncontrolled proliferation, that means this gene has a potential to become an cancer causing gene. Okay, so such genes which has the potential of becoming a cancer causing genes, those genes are known as proto-oncogenes. Okay, oncogenes are the cancer causing genes. Proto-oncogenes are the genes which has the potential of becoming a cancer causing genes. And how this potential will come? Because of some kind of mutations. For example, in genes which are responsible for proliferation, if those genes gets mutated, what will happen? Uncontrolled proliferation will happen. So those genes are your proto-oncogenes. For example, um, um, very famous example is P53. Okay, P53 is also a proto-oncogene. So I think So her question was that short limb is associated with mutation in KITG. So I don't think that short limb is associated with mutation in the KIT because KIT gene is basically a proto-oncogene and short limb is not a cancerous condition. First of all, second of all, KIT gene is mostly observed in skin-related diseases, skin-related cancers. Okay, so I don't think that short limb would be caused because of the KIT gene. According to my knowledge, if there is any exception, I think I would be more than happy if you can come up, if there are any exceptions, if you come up with any examples, then we can discuss it in the subsequent class also. Okay, so today we have discussed about, for the students who have joined very late, so hello, I'm Apurva, and I'm doing PhD in University of Hyderabad, and I'm a PMRF scholar, and that's why I'm teaching you here as a teaching assistant. So the general workflow or the general way of my teaching would be that every week I'll have some 10 to 12 questions which would be related to that week's topic. So like this week I took questions from week 1. So for the next week I'll take questions from week 2. And while we'll discuss the questions, we'll also try to learn the concept that is asked in that question. For example, today we studied about fate map, and also we studied about homologous and analogous structures, and then little bit here and there about some small terminologies or whatever it was there. So like this, we'll try to study the concepts by solving questions, because I believe that if I just start murmuring all and each and everything what is already given in the lectures, in the recorded sessions, then you will just sleep. You will not listen. So it's better that to keep your mind awake, to ask questions, and then with that, we'll try to solve the problems. And again, my request that please try to come on time. Uh, I think discipline is one of the most important and a basic thing, right? So, yes, Namanita, you can ask. You have raised your hand.
Ma'am, I had a doubt from the assignment.
Okay.
There's a question, last question, which of the following morphogenic processes is a key step in forming heart tissue? So can you please
So,
help me with that
actually,
question?
I'm sorry. I'm really sorry, but we are not allowed to discuss the recent assignment questions. Okay. So, I can explain the concepts, but if you are asking me a question, then because we record this, these sessions are recorded and these will be given to the NPTEL team. So it's a strict order that we cannot discuss the present assignment questions. Okay, but we can. Okay, some
Okay,
students
ma'am.
are still joining, but we can always discuss the concept. So can you tell me the concept that you're trying to address? Which concept you're trying to address? The one from the table?
Ma'am, ma'am, the formation of heart tissue. Yes, ma'am. Even if you show me the part where the concept is explained, that then also it will help a lot.
Okay, actually, I don't know the question, so I cannot exactly say that. But I think whatever questions are given, it would be from the PDF itself. So I don't know the question, and it would be better if I you do not tell me the question also. But I think you can go through this table. This table might help you to answer the question. Uh, that's all I can say, because formation of heart tissue, I don't know any topic which we have studied apart from the table here. So I think you can go through the table. Okay.
Okay, ma'am. Okay, ma'am. Okay, ma'am.
Okay.
Thank you, ma'am.
Okay. So Ajay has raised a question. Yes, Ajay, you can ask your doubt.
Recent and please only, madam. But the question number 4, which of the layer will give rise to formation of spinal cord?
Hmm. Okay.
Unicate, I think, don't have spinal cord.
I cannot. So again, I cannot. I mean, we're not allowed to answer, discuss
No,
this.
you don't have to answer, madam. But does tunicate have spinal cord? Tunicate don't have spinal cord. So which layer is going to form a spinal cord?
I don't know what is your
Tunicate.
question. Is it related
Question
to
number 4
unicates?
is, if the figure is given and they are marked ABCD, which layer will form the spinal cord in tunicate? That is the question.
So if you think that
And
the
there is
question
no
is,
option.
if you think that the question is not correct, it is not right, then I think you can. There would be some sessions where your instructor will come and he will also try to address your problems. So I think if you think the question is incorrect, you can try to address the same with the instructor, because he has only made the questions. I have not made the questions. And secondly, just a small recapitulation, that nerve cells, brain, and parts related to this are formed by ectoderm. Mesoderm forms heart, blood, kidney, and bones, gonads, facial muscles, these things. And endoderm forms intestine and lungs, kinds of things. So you can try to relate. I don't know the question exactly, but you can try to relate with it. So generally, your nerve cells and brain, they are formed from your ectodermal layer. Okay, so I think just a minute.
Madam, can I comment on that KIT gene with the question you had answered to Shivan?
Yes.
Actually, there was experiment during piebaldism. Piebaldism, I don't know how to pronounce it. PIEBA, baldism, piebaldism, or piebaldism. The rats were found to have short legs and short limbs.
Okay.
Most of them have dwarfism, and most of them are died. So actually,
Okay.
they had to test it for 3, 4 generations. That's why I think Shivani has asked whether it gives rise to short limb.
Okay. So yes, see,
In
so
the
that
other
is what I'm
experiments, they
saying.
had given
Yeah.
they had given short because it is related with the tyrosine kinase, and that's what you had told us also. It is
So
oncoprotein. So
yeah.
gene involved in growth cell can easily give anything short of the what is normal.
Okay. So it's a I think Shivani, okay, Shivani. So I think there is one experiment which shows that KIT gene is involved in short limbs. In general, KIT gene is an oncogene, but see, science is all about exceptions. There is one experiment that shows. I think if you can also go and read about it, what Ajay has said. So now we'll stop the class here, and we'll meet in the next session. And in the next session, we'll cover the week 2 questions. Okay. So bye, everybody.
Thank you, ma'am.
Okay. Bye.